Hello friends, in this video, that is in the starting of this subject biochemistry, what very easy and good thing I'm going to give you is formulas. Yes, formulas. We'll be doing some formulas which are created by me. So I have given them my name, Smile Formula 1 to 4. Four formulas I'll be discussing, which will help you a lot in making connections in all the biochemistry, in solving so many questions, and especially in regulation. These formulas will help you do the conceptual thing and not cramming, right? And after this video, my next video is how to use the formulas. So in that video, I'll be taking questions and I'll be actually helping you and telling you how you can use these four formulas in solving n number of questions, right? So in this particular video, we are going to take these concepts and basically formulas. Let me tell you, these formulas will be having exceptions also, right? So understand these formulas carefully. Once you understand these, you will be having such a benefit that so many things will be covered with these formulas, right? So let's get started. So coming to formula 1, I call it Smile Formula 1, right? As it brings smile to the students' face, because during question solving, these are really gonna help you, right? So Smile Formula 1 is this, that which pathway is anabolic and which pathway is catabolic. So before starting with the formula as such, let me tell you one basic point in the starting, that we have three states: fed state, fasting state, and starvation state. You know, fed state means we are taking food. Mainly we take carbohydrates in our diet. Within 2 hours of food intake, that period, that duration is considered fed state. Fasting or starvation means lack of food. We are not eating food. But what is the difference? That fasting is of shorter duration and starvation is of longer duration. Let's say like 12 to 18 hours period in which somebody has not taken food since last 12 to 18 hours. So that will be fasting. You know, daily we do fasting during night. You know, you take dinner at around 8 p.m. You take next day breakfast at around 8 a.m. or 9 a.m. So around 12 hours are covered when we're not taking food. So this is also a daily fasting which we are doing. That's why the first meal of the day is known as breakfast. Break the night fast, right? So everything is conceptual if you see it like that. Okay, now what is starvation? Around 1 to 3 days, that duration is starvation. Practically, there is overlap between fasting and starvation. Things are not very much distinguished, but as per theory, we have to make little bit distinction so that you are clear and you are able to solve few questions, right? Like that. Okay. Now, this is one concept, fed, fasting and starvation. On the other side, I will write anabolic or anabolism and catabolism. Now, anabolism means synthesis, right? Synthesis of something in the body. On the other hand, catabolism is breakdown of macromolecules, right? Now, can you connect these two things? Can you connect fed, fasting, starvation with anabolism, catabolism? Can I say that in fed state, body will be in anabolism? Please consider full body as such. We are considering practically the full complete body, human body. When body is in fed state, we are putting the raw material in the body, raw material, that is food, right? We can say body is made up of food, right? We put raw material. So body will be synthesizing cells, proteins, things will be synthesized. So body is in anabolism, right? But during fasting and starvation, we are not putting the raw material in body. So body will be breaking down the stored things for energy, right? So body will be in catabolism, right? So you have connected these two things, that in fed state anabolism will occur, in fasting or starvation catabolism will occur. Now coming to the formula, that which pathway or enzyme is anabolic and which pathway or enzyme is catabolic. First, let's list down the anabolic pathways. Number 1 is HMP, hexose monophosphate pathway, also called pentose phosphate pathway. This is anabolic because this pathway is synthesizing two things in the cells. One is NADPH, other is ribose 5-phosphate, right? So obviously this pathway is occurring in fed state in the body. It is anabolic pathway, right? Then synthesis of glycogen, glycogenesis, synthesis of glycogen, that is also anabolic pathway. You know that whatever excess glucose is there from diet, that is stored in liver so that later it can give glucose to blood. It is also stored in muscles, right? So it is anabolism, that when we have excess raw material, excess glucose, then it is stored in body. Mainly glycogen is stored in liver and muscle. Some amount is also stored in brain, right? So right now we are concerned here that glycogenesis is anabolic pathway. Then any kind of fat synthesis will be obviously anabolic. Fatty acid synthesis, triglyceride synthesis, cholesterol synthesis, that will be anabolic. Then one enzyme which is anabolic is lipoprotein lipase. See, the name is lipase, means lipid breaking enzyme, but I am saying it is anabolic, not catabolic, because when I am telling you all these lists, I am not concerned with one particular thing. I am concerned with body, full body we take practically, that body is in anabolism or catabolism. At that time this thing is occurring, means that is anabolic, right? So lipoprotein lipase detail I will be telling you on next page, but first let's list down all the things in anabolic and catabolic. Coming to catabolic. In catabolic, first is glycolysis. Glycolysis is breakdown of glucose into pyruvate. So breakdown means catabolic. Then link reaction is catabolic. Link reaction is one step reaction after glycolysis, and this reaction lies between glycolysis and Krebs cycle. That is, what is this reaction? The end product of glycolysis, pyruvate gets converted to acetyl-CoA. Pyruvate is 3-carbon compound which gets converted to acetyl-CoA, which is 2-carbon compound. So a 3-carbon compound gets broken down to 2-carbon compound. So this is link reaction, this is catabolic, right? Then breakdown of glycogen, glycogenolysis is catabolic, obviously, breakdown of glycogen. Then beta oxidation of fatty acid, which is catabolic, that is breaking down fatty acid. You know, beta is the major pathway for fatty acid breakdown. Other minor ones are alpha and omega oxidation, right? The next catabolic is gluconeogenesis. Now, be careful here, understand this. Definition of gluconeogenesis is synthesis of glucose from non-carbohydrate sources. But I am saying it is catabolic, not anabolic. It is catabolic because gluconeogenesis occurs in fasting or starvation, never in fed state, right? And one more way by which I can explain you how gluconeogenesis is catabolic, understand one basic point, that this gluconeogenesis is making glucose in human body. Otherwise, we don't have to do such a thing. Making glucose is the work of plants by photosynthesis. We animals and humans usually get glucose in our diet, right? Usually, we don't have to make glucose. But when it is a situation in human body that we have to make glucose, which is difficult pathway, high energy consuming pathway, then what is happening in the body is this, that something in the body is broken down to give glucose, right? So in body to something is getting broken down to give glucose, right? So body is in catabolism at that time. When body is in catabolism and at that time this pathway is occurring, gluconeogenesis, so we will consider the body, that body is in catabolism or anabolism. So when body is in catabolism, then we say that gluconeogenesis is catabolic, right? The next is anything of ketone body synthesis as well as ketone body utilization, that will be catabolic, because ketone bodies are formed during starvation. You know, ketone bodies are formed by only one organ, that is liver, to be used by specially heart and brain, right? Heart and brain uses them during starvation, right? So obviously body is in catabolism at that time, so these pathways are considered catabolic pathways. Then the last one is hormone sensitive lipase, HSL, hormone sensitive lipase. Okay, so I am done with the list, but note one point here, that this is also a lipase, this is also a lipase, but I am saying that lipoprotein lipase is anabolic, but hormone sensitive lipase is catabolic. Why? Let's understand this. So let's first take lipoprotein lipase. This enzyme is present in the blood vessels. It is present in the endothelium of blood vessels. So in the endothelium of blood vessel, this enzyme is present, right? So this is blood vessel I am showing here. The sides of blood vessel, we have peripheral tissue, especially adipose tissue I am considering here. So what happens in blood? Lipoproteins are moving, chylomicrons and VLDL. They both contain which lipid? They contain triglyceride. They are moving in blood. So here this lipase enzyme, lipoprotein lipase enzyme, will break down this triglyceride. It will break down this triglyceride. This triglyceride will be broken down into 3 fatty acid and glycerol. You know, triglyceride means 3 fatty acid and glycerol is present. So this will be broken down into its constituents. These are smaller molecules. Fatty acid and glycerol are smaller molecules. They can be easily absorbed into the peripheral tissues. They can easily enter the peripheral tissue, not absorbed. They can be entered into peripheral tissues. So here in adipose tissue, they will be rejoined to make triglyceride again, and this way triglycerides are getting stored in adipose tissue, which is very clear that this is an anabolic role in the body. It is anabolic role in the body, that whatever fat, whatever lipids are moving in the blood, they are transferred from blood into the adipose tissue and they are stored in the adipose tissue, right? Obviously such kind of thing is occurring in fed state in body. So this lipoprotein lipase enzyme is anabolic enzyme. Also focus on the name of this enzyme. It is lipoprotein lipase. It is breaking the lipid lipase. It is breaking the lipid of lipoprotein. Let me be very clear. Chylomicron and VLDL are lipoprotein and triglyceride is lipid. More clarity you will get when you will watch the video on lipoproteins, right? And now coming to other enzyme, hormone sensitive lipase. Hormone sensitive lipase. This enzyme is present inside the cells of adipose tissue. I told you lipoprotein lipase is present in the blood vessels, but this enzyme is present in the cells of adipose tissue, and it is catabolic enzyme. When body is in catabolism, then body wants to use the fats of adipose tissue. Fats of adipose tissue are triglycerides. They are broken by this enzyme, hormone sensitive lipase, again into fatty acid and glycerol. These fatty acids will go into blood. From blood, they will be taken by liver, where beta oxidation of fatty acid will occur, right? So hormone sensitive lipase is catabolic enzyme. It is breaking the lipid of adipose tissue, right, during catabolic situation in body. So with this we are done with formula 1. Now coming to formula 2. So again it will be SMILE formula 2, in which I will be telling you which hormone activates which pathway, or which hormone is anabolic and which hormone is catabolic. So first the basic here, that insulin is always anabolic hormone. The other opposite hormone, glucagon, is catabolic hormone. You know that we have mainly these two hormones working in the body, insulin and glucagon. So insulin is anabolic and glucagon is catabolic, right? Insulin is always anabolic. It will store all the things in the body. It will not allow breakdown. You can note this point some way practically. If you see some of your patient or neighbor or relative taking insulin injections since last few years, then these patients will be having large abdominal circumference, because they are taking insulin, and insulin will store all the things in the body, not allowing breakdown. So because it is anabolic hormone, so taking insulin will increase weight, obesity, and abdominal circumference will be raised, right? Now how insulin is anabolic? If I tell you like very simple basic concept, you think here, you know, when we are in fed state, we mainly take carbohydrates in our diet, mainly which is glucose. So I can say that in fed state, glucose is entering our blood and hyperglycemia is going to occur, but insulin comes here to decrease blood glucose. So in fed state, insulin is released. If insulin is coming in fed state, means insulin will be anabolic hormone. Insulin is always anabolic. So done with the basic concept, let's take the formula. So the formula is that insulin activates all anabolic pathways and enzymes, especially the regulatory enzymes of the anabolic pathway will be activated. Anabolic enzymes like lipoprotein lipase will also be activated by insulin. So there is a direct clear-cut relationship here, right? That insulin activate all anabolic. I've already told you in Formula 1 which is anabolic, which is catabolic, right? So use that knowledge for combining now Formula 1 and Formula 2, right? So insulin activate all anabolic pathway and enzymes. But I told you, we have exceptions. You have to take care of the exceptions also. So the exception is, but insulin activates two catabolic pathways also. Insulin activates all anabolic, also two catabolic are activated. Which are these? These are glycolysis and link reaction, right? So insulin will activate all these. So the exact opposite formula, very easy to write. The opposite hormone, glucagon. Now glucagon activates, so you can tell all the things. Glucagon activates all catabolic enzymes and pathways. But what is the exception? Same exception. But it will not activate two catabolic, that is glycolysis and link reaction, because these two catabolic are activated by insulin, right? So I repeat the formula once. Smile formula 2 is this, that insulin activates all anabolic pathways, plus it also activates two catabolic, that is glycolysis and link reaction. What is link reaction enzyme? Link reaction enzyme is pyruvate dehydrogenase complex, right? Then opposite formula is glucagon activate all catabolic pathway and enzymes, but it will not activate two catabolic pathways, that is glycolysis and link reaction, right? So with this we are done with formula 2. Coming to formula 3. Smile formula 3 is about phosphorylation and dephosphorylation. You know, questions are asked that which enzyme is active in phosphorylated state, which enzyme is active in dephosphorylated state, which hormone causes phosphorylation, which hormone causes dephosphorylation, what is cyclic AMP. All these things will be solved under this heading of formula 3. So let me tell you, in short, insulin always causes dephosphorylation, and obviously you know for the other one, opposite, glucagon always causes phosphorylation, right? And insulin decreases cyclic AMP and glucagon increases cyclic AMP. Let me tell you, there is always an inverse relationship between glucose and cyclic AMP. Look here, in fed state we have more glucose, so we have insulin, so cyclic AMP is decreased. In fasting state we have glucagon, which will increase cyclic AMP, but at that time glucose is less, right? Now let's take the detail of this cyclic AMP. Cyclic AMP is a second messenger which activate protein kinases. It activates kinases. Kinases will do phosphorylation. Various enzymes or proteins are getting phosphorylated. So in short, can I say cyclic AMP causes phosphorylation, right? Now, cyclic AMP is synthesized from ATP by enzyme adenyl cyclase. Adenyl cyclase. You know, very easy to learn the name of this enzyme. ATP gets cyclized, cyclic AMP. So, enzyme is cyclase, adenyl cyclase. And cyclic AMP is destroyed when it is broken down, then it gets converted to 5-AMP, and here enzyme is phosphodiesterase, right? So I have given you two enzymes here, adenyl cyclase and phosphodiesterase. You will tell me that insulin activate which enzyme? Insulin activate which enzyme? Insulin activate phosphodiesterase. So insulin will decrease cyclic AMP because insulin will convert more of cyclic AMP into 5-AMP, and therefore when cyclic AMP is not there, then insulin will cause dephosphorylation, right? So insulin decreases cyclic AMP, insulin causes dephosphorylation. On the other hand, hormone glucagon will activate adenyl cyclase. This way, glucagon increases cyclic AMP and causes phosphorylation. Even other hormones, catecholamines, epinephrine, norepinephrine, growth hormone, glucocorticoid, and even thyroid, they all increase or activate adenyl cyclase, and therefore they all cause phosphorylation, right? But mainly here I am saying glucagon. So coming to the exact formula number 3 is this, that the pathways or enzymes which are activated by insulin are always active in which state? Dephosphorylated state. I told you insulin causes dephosphorylation and insulin activate anabolic pathways. So basically now we are combining all the three formulas and making a concept that all the anabolic pathways plus glycolysis link reaction enzymes, they all will be active in dephosphorylated state. So I repeat, the enzymes and pathways which are activated by insulin, they all are always active in dephosphorylated state. Let's write down the exact opposite of this. The pathways or enzymes which are activated by glucagon, are always active in which state? You know that? Exact opposite. They are activated in phosphorylated state. And you know which are these pathways? These are catabolic pathways, but do not consider glycolysis and link reaction here. You already know that exception, right? So this is done, that anabolic pathways are active in short, anabolic pathways are active in dephosphorylated state, and most of the catabolic pathways are active in phosphorylated state. But we have one exception here. There is one enzyme, ATP citrate lyase, which is anabolic enzyme because it is involved in citrate shuttle of fatty acid synthesis. So very clear, fatty acid synthesis enzyme means it is anabolic enzyme. This is anabolic enzyme. Usually anabolic enzymes are active in dephosphorylated state, but this anabolic enzyme is active in phosphorylated state. Only this one exception you have to learn. If you are getting confused, you can skip this, because if it is a very difficult exam, then only they'll ask for this very rare exception, right? Otherwise, just consider the normal thing. Anabolic enzymes are active in dephosphorylated state. Catabolic enzymes are active in phosphorylated state, right? If you're getting confused for this particular ATP citrate lyase, you can just skip it, right? Coming to the last formula, fourth formula, formula 4, SMILE formula 4, in which we will be doing with the concept that which pathway occurs in which compartment of the cell. You know that few pathways occur in cytoplasm, few occur in mitochondria, and in books when they write, they do not tell you any concept, right? You have to mug up this thing, which becomes very difficult when you are revising, right? So I will be telling you this thing with the concept, and let's understand this very clearly. You know which are anabolic, which are catabolic pathways, right? Now look here. Anabolic pathways occur in cytoplasm, right? And catabolic pathways occur in mitochondria. Very easy. Anabolic in cytoplasm, catabolic in mitochondria. So when a cell has to break something, cell will use mitochondria to break that compound. And when a cell has to make something, cell will use cytoplasm to make that thing, anabolism, right? So very clear, fatty acid synthesis will be occurring in synthesis, anabolism will be occurring in cytoplasm. On the other hand, beta oxidation of fatty acid breakdown will be occurring in mitochondria, right? So this is short formula, but we have exceptions to this formula. Two exceptions: glycolysis and glycogenolysis. Glycolysis and glycogenolysis both are catabolic pathways. According to our formula, they should occur in mitochondria, but no, they both occur in cytoplasm. So these are two exceptions here, right? Be careful. Glycolysis and glycogenolysis are catabolic, but they both occur in cytoplasm, right? Now one more thing, that few pathways occur both in mitochondria and cytoplasm. Three here you have to learn: gluconeogenesis, urea cycle, and heme synthesis. All these are used when these three pathways occur. Both mitochondria and cytoplasm are used. How to learn this? Can you see something common in these three pathways? Can you see something common? Think. Making glucose, making urea, making heme, making these three compounds is very difficult. I'm just giving you a hint so that you can easily learn these three. Making glucose, one molecule of glucose requires 6 ATPs. Making urea, 4 ATPs are used. Making heme is such a complex compound, 4 rings with central metal, very complex compound. So when these three difficult things a cell has to make, then the cell will take the help of both the compartments, mitochondria and cytoplasm, right? So easy to learn this way. And one more very good thing, that whenever a pathway occurs both in mitochondria and cytoplasm, then that pathway starts from mitochondria. First, one or two steps occur in mitochondria, right? So, very easy to answer that. Sometimes they ask you first enzyme of these three pathways. First enzyme of these three pathways lies where? Very easy. These three pathways, first enzyme will be in mitochondria, right? So, we can also see one thing here like this, that gluconeogenesis is also an exception I will consider. Gluconeogenesis, I told you, is a catabolic pathway. Catabolic pathways usually occur in mitochondria, but this catabolic pathway occurs both in mitochondria and cytoplasm, right? Then I have not considered, I have not taken TCA, ETC, these two pathways I have not taken in my list of catabolic, anabolic, because these two are vital pathways and they do not depend on hormones. They do not depend on fed or fasting situation. They do not depend on whether we are taking food or not taking food, because these are vital pathways. They are occurring as per the vitality of the cell, right? They do not depend on our food status, right? So these pathways, vital pathways, let me tell you, vital pathways also occur in mitochondria, right? So with this, I have completed the formulas. I hope these formulas will help you a lot in solving questions. So you can watch how to solve questions in my next video, right?